So the story of evolution goes on. Once we have multicellularity and the appearance of true animals, we have an explosion of different types of animal body plans. This explosion took place about 540 million years ago during the Cambrian period. But the first animals are believed to have existed at least 600 million years ago in the Ediacaran period. Of course, such diversification of body plans could only have taken place if genomes became more and more complex, with genetic innovations that allowed all this experimentation of biological solutions to fill in new ecological niches. Again, the inspection of genomes of present-day animals, similar to those first animals, will help us to have a pretty good picture of what may have happened. The classical tree of the first animals is usually thought to be something like this, with sponges, porifera as the most basal group. Then we would have comb jellies or ctenophores, then jellyfish, also called cnidarians, and then all animals with bilateral symmetry, that is, all the rest of invertebrates and vertebrates. This tree was built according to some biological features, whether there is a nervous system or not, the type of symmetry, the presence of muscle cells, etc. But in recent years, scientists have sequenced the genomes of sponges, of comb jellies, and also the genome of another interesting creature that lies somewhere in between. And the analysis of these genomes, you should expect it by now, gave us quite a few surprises. Trichoplax adherens is the only species belonging to a group called Placozoans that looks very much like a sponge. It is like a ball of cells that lives in fresh water. Like sponges, Trichoplax lacks nerve cells, of course, but since their cells are joined by desmosomes, a specific type of structure that keeps cells together, and desmosomes are not present in sponges, people think that Placozoans, Trichoplax, evolved later than porifera. The Trichoplax genome, sequenced in 2008, is 98 megabases long, twice the size of the genomes of a typical choanoflagellate, but still has a number of protein-coding genes, 11,500, very similar to the number of genes in choanoflagellates. But what is interesting about genes in Trichoplax is that they have a structure which is remarkably similar to the human genes. And what is really amazing is that the genome has very old versions of at least one homeobox gene. And as you probably know, homeobox genes are a large class of genes involved in specifying the identity of body segments during embryonic development in complex animals. This family of genes is present in all animals with bilateral symmetry, but what are they doing in an animal which is basically a ball of cells with no head or tail, with no symmetry at all? Well, we might get an answer by looking at the genome of sponges. And the first sponge to have its genome sequenced, this was in 2010, was Amphimedon queenslandica, a demos sponge from the Great Barrier Reef in Australia. The genome of this sponge is larger, 167 megabases, and has a surprisingly high number of genes, about 20,000 genes. Again, these genomes show some advanced features, transcription factors, homeobox genes, adhesion molecules, and signaling pathways necessary for a multicellular life. What is really amazing is that the sponge, which of course lacks neurons, has quite a few of the genes that are used to form synapses, the connections between nerve cells. In this case, scientists think that they know what is going on. Apparently, these genes do not work in a coordinated fashion in the sponge. It is precisely that coordination which is required for the correct formation of functional synapses. So it seems that the connections between nerve cells evolved by recruiting genetic components and getting them to work together in new ways. Yet another example of co-option and acceptation. So one could say that the ability to have a nervous system was already implicit somehow in animals that had no brains. However, the final word in this area has come from a different type of animals, the comb jellies or ctenophores. These beautiful bioluminescent animals have biradial symmetry and some primitive nerve cells. In the classical tree, ctenophores were placed later than sponges and placozoans, but just at the end of the year 2013, we could have a look at the first ctenophore genome, and what we have learned from this is really changing our ideas about the early evolution of animals. Mnemiopsis lady, also known as a sea walnut, has a genome which is 155 megabases in size and contains 16,500 protein coding genes, a bit smaller than the genome of the sponge and Phimedon. Scientists were very surprised to find a striking pattern. The co-inheritance here means the shared presence of genes in both sponges and comb jellies. The co-inheritance of two types of genes for some transcription factors and genes that are important for the development of a nervous system in general. This suggests that the genetic machinery required for nervous system development might have been present already in the ancestor of all animals. In ctenophores, comb jellies, this would have led to the formation of nerve cells, whereas apparently this ability to form neurons or neural systems was lost in sponges, even though they have the genes. However, although comb jellies have a very primitive type of nerve cells, they do not use a neurotransmitter called serotonin, which is a universal neurotransmitter in the rest of animals. So the ability to form neurons might have evolved independently twice. In fact, typical neuronal networks of bilateral animals will not appear until much later in cnidarians, the jellyfish. So when scientists built phylogenetic trees, including the genomes of comb jellies, Trichoplex, and sponges, the tree that came up as the more plausible one had ctenophores as the most basal group. The nearest to the root, so to speak. So this is illustrated, for instance, by the analysis of microRNAs. That class of small RNAs that are so important in the regulation of the activity of many genes. Some animal microRNAs are found in sponges. The genome of the sponge and Phimedon contains at least 8 microRNAs typical of animals, as well as all the genes necessary for the biosynthesis of microRNAs. But neither ctenophores nor placozoans seem to use microRNAs because you can't find any known animal microRNA in the genome of Mnemiopsis or in the genome of Trichoplex. In fact, they don't even have the gene for a protein called PASHA, which is crucial for the biogenesis of microRNAs. So it is things like this that support a more basal position of comb jellies than sponges in the phylogenetic tree of animals. All this, of course, is provisional because we are just starting to get the first genome sequences of species from these groups. Considering all that we have learned by reading the genome of just one sponge and one comb jelly, if you think that many of the genomes might be completely sequenced over the next couple of years, you will agree with me that these are really exciting times in the field of comparative genomics.